Hello. Okay, we're not going to get started yet, but I'm just making sure. Can you guys hear me right now?
Yeah.
Okay, good. Woo. All right. Well, do we want to go ahead and get started, or we want to take just a few seconds for everybody to get here? That's kind of what I'm thinking. What do you think, Manoj?
Yeah, we can wait a little while for everyone to get on.
Let's get everyone. I don't know if people are just starting in this session and weren't in the previous session. We ran a little bit over, so we'll give everyone a little, and then we'll get started with the research roundtable. I'm going to go off camera, so I'll see you guys on the other side.
Sounds good. While we're in here, let me go ahead and just kind of give everyone an update on the expo. So remember that in our expo, we have the lovely videos from our SCN8A families. And then we also encourage you to check out the section of Dr. Hammer's video. He has the featured presentation on sex and strain differences in the natural history of the SCN8A in 1768 mouse model. When we asked Michael what he wanted to present on, he wanted to be certain to reach the research community, to let them know his willingness to share his mouse models on different genetic backgrounds that exhibit a broad phenotype spectrum. In order to give him plenty of uninterrupted time in this tight schedule and to share his research, his presentation is now available and will be for the duration of this two-day event in the expo section. So, and then it will also be on our website after that. So please know that the expo, we've actually extended that, so it will be open through Sunday. That way we have plenty of time to go enjoy the kids' videos, and we have plenty of time to look at Dr. Hammer's video and be able to absorb all that information. So, all right, and guys, we ran a little bit late on the last session, so we were going to kind of maybe hang out here for just a few minutes and then get started. So I'll go ahead and hand it. Oh, actually, we're going to wait because I forgot that we are going to show a video coming up. So let's hang out for maybe 2-3 minutes, and then we'll get that video started. Does that sound good to you guys?
Sounds great.
Perfect.
Milo is a motivated 17-month-old boy who has endured more than most would in their lifetime. His seizures began when he was 3 months old, but they never stopped him from smiling and giggling every day. He loves to play with his feet, show off his new skills, eat his vegetables, even asparagus and artichoke, and take in all the sights and sounds during his daily stroller rides. Nothing could prepare him for the latest seizure cluster, status epilepticus, and subsequent 25-minute cardiopulmonary arrest, which led to severe brain damage, from which he may not fully recover from. Milo is one of only several hundred children globally who are diagnosed with SCN8A related epilepsy with encephalopathy.
SCN8A is a gene that encodes the sodium channels NAV1.6, a protein on the surface of brain cells that is necessary for normal brain activity. When this protein is mutated, it impedes the cells from properly responding to an action potential, commonly through faulty inactivation, causing a gain of function. Mutations in this gene can cause neurological problems, including epilepsy and learning difficulties. The link between SCN8A and epilepsy was discovered in 2010, and due to advances in genetic testing, more children are being diagnosed. It is estimated that 1 in every 55,000 births will be diagnosed with an SCN8A variant, or approximately 1% of all epilepsies.
The Cute Syndrome Foundation's mission is to raise awareness of SCN8A mutations, to fund the dedicated and talented scientists researching SCN8A, and to support the families around the world who are affected by this disorder. Since 2010, over 100 research articles have been published on this disorder, 25 of which were published in 2019 alone. As research labs and clinicians continue to expand our understanding of SCN8A and develop a cure, more funding is needed to help Milo and his extended SCN8A family across the globe. Hilary Savoie, the founder of the Cute Syndrome Foundation, is the cog that keeps everything moving forward. She orchestrates multiple teams to achieve the same goals. She is not alone. Shelley, Casey, and many others all form this engine, as do the parents, the children, and all those who study and care for them. Milo, these words are for you, even though you cannot comprehend what I say. You feel it through our love and hopes. I hope you never lose your sense of wonder. May you never take one single breath for granted. I hope you still feel small when you stand beside the ocean. Whenever one door closes, I hope one more opens. I hope you never fear those mountains in the distance. And when you get the choice to sit it out or dance, I hope you dance.
Absolutely beautiful, Gail. We were hesitant to ask her for a video given what they've been going through, but we were shocked to receive this stunning video submission. So thank you for doing that, and all of our hearts are with you guys as you guys go through this new normal. Now we'll move on to the researcher roundtable. Remember, if you have any questions, please type them in the chat box to your right, and I will hand it over to Dr. John Schreiber to get us started.
Thank you. So, Manoj, I didn't know if you were going to introduce this section.
No, you can do it. That's fine.
All right, let me see here.
If you've got it. Well, I was going to say
I don't have the schedule in front of me.
Yeah. So what I was going to say was, so we have a great set of talks coming up now, and we're going to start with Dr. Miriam Meisler. She's going to be talking about her antisense in the SCN8A. So we'll go ahead with Miriam. Miriam, you are we muted you, so if you could just unmute yourself again. Please, that's it. And then we're going to get Miriam to go first, and Miriam's going to be followed by Carlos, and then she's going to be followed by the two guys from my lab, Ian Wenker, and then Jeremy. So we're going to start with Miriam, and the title of her talk is Antisense Oligonucleotide ASO Treatment in SCN8A.
Okay, thank you very much, Manoj, and I'm so glad to be here. I'm going to revisit a topic that I talked about last year at our meeting because I think the ASOs have so much promise for the future, and I'm not sure if they've been covered already today. But I'm basically going to review how the ASOs work, and especially for SCN8A. So the next slide shows what I think everybody knows today, and that is on the left that DNA is a double-stranded molecule, and it's held together by hydrogen bonds between complementary bases. It's very, very stable. We can separate those strands with heat, for example, but if we then cool the solution back down, they will find each other and bind exactly as they did before, because of the stability of that structure. Now, on the next slide, you can see that the same thing is not only for long DNA molecules, but for very short DNA molecules. So these little 20 base pair, little pink molecules are what ASOs look like. They're tiny pieces of DNA, but they have a complementarity to the big piece in the gene or the messenger RNA. So if we, again, separated the two strands of the DNA and then cooled it again, these very small molecules would form stable dimers with the DNA. And so that's the basic principle of what's happening with ASO therapy. So on the next slide, you can see the remarkable fact that these antisense ASOs are taken up by neurons very readily. So what we have to do for therapy is get them into the cerebrospinal fluid. Once they reach that fluid, the neurons will take them up, and in the neuron, they can hybridize either in the cytoplasm or the nucleus. So for the point of view of SCN8A, the critical hybridization is in the cytoplasm. The ASO finds the messenger RNA for SCN8A, and that's the target we're after. And the reason this is helpful is that there's an enzyme in the cells called ribonuclease H, or RNase H. And this enzyme recognizes this double-stranded molecule and recognizes that it's a foreign molecule and degrades it. So that's the goal for treating SCN8 encephalopathy. It's to reduce the amount of this messenger RNA using the ASO. So on the next slide, there's just a summary of what you all know about SCN8A developmental and epileptic encephalopathy. And you know that SCN8A encodes the channel called NAV1.6. And the predominant molecular mechanism, as you've been hearing, is, first of all, the mutations are de novo. They're not inherited from parents. They are missense mutations with a substitution of one amino acid for another. And in general, they are gain of function mutations, meaning that the protein is present, but it has abnormal properties in terms of sodium channel activity. So the next slide, you see 3 examples of these gain of function mutations and what they do to the channel. The mutant channels are in red. And you can see on the left, this is a representation. The black dots show you how a normal channel should open as your neuron is getting ready to fire an action potential and to be active. But this red channel is opening too soon, and therefore you're getting excess hyperactivity in the neurons if you have that mutation. The panel in the middle shows another mechanism. These channels are failing to close properly. So while the black channel is closed within a few milliseconds, these mutant channels take longer to close. And again, that makes the neuron more active and continue to fire action potentials when it should stop. And I should mention this is work from Manoj Patel's laboratory several years ago. And on the right, you see another abnormality. This particular channel opens okay, it starts to close, but then it fails to close completely. So that out here, when the wild type channel is nearly completely closed, we have an enormous amount of current flowing again. And again, this is making the neurons more predisposed to fire and to be active. So on the next slide, you see an example of this hyperactivity in neurons. Now, these are neurons. We have put some of these gain of function mutations that are found in patients. We have made mouse models that have these same mutations. And here's what you can see in this particular model. On the left, you have recordings from hippocampal neurons, CA1 neurons, CA3 neurons. These represent 10 seconds of recordings. And you see these neurons are completely inactive when the cells are taken from a wild type mouse. But on the right, you can see that when you take the same neurons from the mutant carrying this patient mutation, they're firing inappropriately. They're more predisposed to fire because of that failure to close the channel completely. So this is a representation of the problem of from these gain of function mutations in SCN8A. So the next slide, please. Thank you, Shelley. So this is a paper that we published in February showing that we could use an antisense oligo in these mouse models, and it would be protective, not only for SCN8A encephalopathy, but also for the related Dravet syndrome. So the next slide, and I should mention, this is work from our lab and in collaboration with Ionis Pharmaceuticals. So here's this mouse that we were using. So this mouse is expressing R1872W, which is located in the C-terminal cytoplasmic domain, and it prevents stable closing of the channel. And here you see control mice, and here you see mice that are expressing this channel. And you see they have a very rapid course. This is the percent survival. And in fact, most of these mice die right after their first seizure at about 2 weeks of age. So it's a very severe mutation. Now, in the next slide, I'm just showing you where we made this ASO. This represents the SCN8A mRNA. And we were asking, can we treat this disorder by reducing expression of NAV1.6 using an ASO to start the degradation product? And the ASO is complementary to this part of the gene, messenger RNA. So the next slide, please. The first question was, does this ASO, in fact, reduce the transcript level? So we administered it into the ventricles of the brain in the very young animal before seizures had begun at postnatal day 2. And you see here, we set 100% as the mouse that doesn't receive any ASO. And these mice received 15 micrograms of ASO. They have less messenger RNA, reduced from 100%. And here, these mice received 30 micrograms of ASO. And they're down to 50% of the messenger RNA level. So this demonstrates that, in fact, this works in an animal, in vivo. If we can get this ASO to the neurons, it will reduce the amount of messenger RNA. Now, what does that do for the survival of the animals? That's shown on the next slide. Now we're looking at the same mouse, and we see the treatment of P2 extends the survival. Here's a mouse that received a control ASO that's not related to SCN8A. And it looks just like the mice I showed you a minute ago, 2-week survival, very rapid fall off. And here are mice that received 3 different doses, 15 micrograms, 30 micrograms, 45 micrograms. Now they also do not survive, but they survive a lot longer than the mice that got the control. So eventually they succumb, but depending on how much we injected, they go from 2 weeks to 7 weeks survival, which is 3 times longer. Here you can see that there's a dose dependence. The more ASO, the longer they survive. So why are they not surviving forever? You can see on the next slide, we thought, well, perhaps the ASO is wearing off and we could give them a second dose and they'd live longer. So that's what this experiment is. This is untreated mice. These are mice that got 30 micrograms and they lived for almost. And then there are mice that we injected again, just as they were old enough to start to have seizures. And you see, we got another improvement. We got another period. And I should mention that during this period, when the mice are surviving, they're also perfectly normal by any of the tests that we did. And they're not having abnormal EEGs, they're behaving normal, their movement is normal. So it seems that as long as they remain above a critical threshold, they're really protected. And then once again, we saw the onset of seizures. So the next slide, we asked, are they having seizures because the transcript or the messenger RNA is coming back? And that's exactly what it looks like. Now here is the levels 3 weeks after the treatment, and that's down to 50% in these animals. But after 5 weeks, it's beginning to come back. And when we look at them immediately after they have a seizure, we can see that the transcript is back to wild type levels. So in order for this therapy to be applied, it has to be repeated administration to keep these transcripts low. And on the next slide, I summarize what we found so far. And that is, first of all, that the SCN8A ASO, which goes to the 3' end of the messenger RNA, when administered to the mice, does reduce the messenger RNA in vivo. And our experiments started by administration at postnatal day 2, but we're now trying to treat them later and see if we treat with the ASO after the first seizure, can we prevent subsequent seizures? So the reduced expression in these mice with gain of function, it does rescue the seizures and the comorbidities as well. And repeated treatment extends the effectiveness, but the ASO does need to be administered directly into the brain or the spinal cord by intrathecal injection. And we are looking into possibly getting around this limitation by introducing the ASO into nanoparticles, for example. But this is something for the future, hopefully. And again, it will be very important to regulate the dosage, because we know that if we go too low with SCN8A, we run into different problems, and we don't want that. So that's a status, and I hope it clarifies this approach. And on the next slide, I would like to acknowledge the people who did this work in my laboratory: Guy Lenk, Sophie Hill, and Wenji, and as well as JC and Rosie, and we worked with Frank and Payman at Ionis. And these are the wonderful funders of our research, including the Cute syndrome, for which we're very grateful. So, thank you very much.
Thank you, Miriam. That was an incredible talk.
Thank you, Miriam.
Phenomenal work. So we have some questions coming up. I think you answered one of them, and that is lipid nanoparticles may be used in the future to deliver the ASO. But do you think alternative gene therapy approach like RNA editing is a viable way to go forward as well?
RNA editing, meaning using CRISPR.
Yeah.
Specifically, we're hoping that this approach, the ASO approach, will work on many different mutations. And when you get to editing, you first of all need a whole different set of reagents for each mutation. So that immediately makes it more difficult. And you need CRISPR. And right now, CRISPR has not been applied to human in any case that I know of. And I think that is a way down the pike, at least. Well, I won't guess how long. Maybe you could all guess how long before CRISPR becomes feasible in human. So the CRISPR system could be used to correct these mutations, but those are the limitations right now.
Okay. We had another question from Tyler, and it says the initial experiment indicated that the repeated administration did not have an effect as long as the initial injection, i.e. the 30 micromolar, had more than the second injection of 100. Is there a longer data set just to confirm the extension of effects will continue in later doses?
Yeah, we're doing that right now, and in fact, we could increase that 100. See, the brain is growing during that period of time, and you need more ASO to reach the whole brain. And we're going to try 500. I didn't mean micromolar. Goodness.
No, I think it was what you had in the.
Yeah.
I just want
So we do think that a higher dose might persist longer, will persist longer.
So I had a quick question, and that was, do you think if we started specifically targeting the different neuronal types, you might see a longer effect? Because I guess the ASO is not distinguishing which neurons it goes into excitatory, inhibitory, right? And I'm just wondering if you're now kind of changing that balance. And so what are your thoughts of that, Miriam?
Well, from the behavior of these mice, it does not seem to be necessary. All the neurons, the inhibitory and the excitatory, in fact, are expressing all of these sodium channels. And it's more deleterious in some, depending on the whole balance of channels. But I don't think it's going to be necessary to take that further step.
I think in the interest of time, we'll probably move on to the next talk. Thank you so much, Miriam. That was a wonderful presentation. We'll continue now with Carlos Vanoy. I think I'm going to, sorry if I mispronounced your name. I don't think we can hear you, Carlos. We can't hear you, Carlos.
Just have to unmute yourself.
It might be that you're on mute, Carlos. I'm not sure if we can get some technical assistance to mute Carlos on instead. Can somebody do that?
Yeah, it's not allowing me to do so.
Do you see a little mic button at the bottom of your screen underneath all the squares? Let's see. Unfortunately, I can't unmute you from my end.
Okay.
He's going to join us again.
Yeah, give him a second. He should, I'd say he'll come back in and see if that helps.
I'm going to say while we're waiting, looking at Miriam's slide from the 1768 mouse reminds me of what Laurie Eisen presented 2 years ago with now what's going through clinical trials now with Stoke, which we're really excited about. And it's just, that's only 2 years ago. I think it's really exciting about how fast it's going to move. Good.
Okay.
We can hear you now, Carlos. Okay. Thank you, Carlos. Go ahead.
Okay, let me see. And I'll be sharing the screen. And let me know when I should start, I guess.
You can start, Carlos. We can see your screen.
Okay, I'm ready to go. Okay. I want to thank you. Good afternoon. I want to thank the organizers for allowing us the opportunity to present some of our work doing functional analysis of adult and neonatal human NAV 1.6 and epilepsy-associated variants by high-throughput electrophysiology. This work has been done in the lab of Al George at Department of Pharmacology at Northwestern University, and the people listed here are all crucial for the continuous progress of this project. And this research has been supported by a Centers Without Walls grant from the NIH. So the purpose of this study is because SCN8A, like other sodium channels, is subject to post-transcriptional and developmentally regulated alternative splicing. This splicing leads to multiple isoforms, and these isoforms may have distinct functional properties. So when we look at the behavior of epilepsy-associated variants, these variants may exhibit distinct properties depending on which splice isoform is used for the functional analysis, like if you use adult or versus neonatal, for example. And the significance of looking into see if we're going to be looking at the variant function using adult or neonatal isoform is illustrated by this figure here from a very recent paper where it shows the expression levels of the adult and neonatal SCN8A throughout development. And you can see that throughout development, the amount of 8A adult versus neonatal changes. So it's possible that when you're looking at the function of the variant, it's important to know what isoform adult or neonatal is being used. So our goals for this study is to compare the functional properties of wild-type adult and neonatal NAV 1.6 isoforms. And we also wanted to test whether disease-associated variants exhibit isoform specific, that is, do they behave differently in the adult versus the neonatal isoform. To do an efficient analysis of ion channels, we have developed a workflow that combines a high-efficiency electroporation. This allows to transfect cells with the desired protein at high efficiency levels where we can do high throughput automated patch clamp. And by able to do high throughput automated patch clamp, we can record from up to 768 cells at a time, which if you compare, for example, to a conventional method, manual patch, you can only record one cell at a time. So, and this is the ability to do it high throughput is very important, because if we look at a recent listing of how many variants in voltage-gated sodium channels are associated with epilepsy, this list exceeds already 2,500. So the ability of being able to do high throughput analysis will allow us to, be able to determine whether any of these variants or all of these variants have any kind of functional defects. This is a screenshot of an ongoing experiment where we're recording sodium currents from a neuronal cell line that we developed in the lab that is modified. It does not express endogenous sodium channels. So when we put our sodium channels of interest, we can, detect it without having to use, other maneuvers to differentiate between the endogenous and the exogenous. Here I'm showing you on the left and right currents recorded from 3 non-transfected cells. These are the currents here on the left and on the bottom, so very small current. And then we're also recording from the adult isoform and the neonatal isoform. So in this plate, for example, of 384 wells, we're able to analyze data from almost 300 cells, which met the minimum criteria that we use for considering a cell usable for data analysis. These are the whole cell currents recorded from non-transfected cells and cells transfected with either adult or neonatal isoform. We can see the nice inward voltage-gated sodium currents. And here on the right, we have the voltage current relationship showing the currents for the non-transfected cell, very small. And when we look at the adult and the neonatal, we see these large inward currents. When we look at the voltage dependence of activation and inactivation, and if we compare the adult versus neonatal in the for this channel, we see that for voltage dependence of activation, there's really no difference in the voltage dependence of activation values. The properties are identical between the neonatal and the adult isoform. However, when we look at the voltage dependence of inactivation, we see there are differences when we look at the neonatal field symbols versus the adult, open symbols, and we see it's a 5 millivolt shift. So having neonatal versus adult, we already see a difference in the behavior of the channel. So following that, we wanted to detect whether if we look at variants in express median in the adult or neonatal, can we detect there are differences that are specific to the adult or neonatal isoform. So here we're showing the currents for the wild type channel expressed in the adult isoform and for 2 epilepsy-associated variants, G1475R, which from here on will be shown in red for those data, and for the mutant N1768B, and from now on it will be shown in blue. And then we also have the current traces recorded from the same variants, but now expressed on the neonatal isoform. So because I'm going to be showing a lot of data from different variants, one way we thought about be easier to explain this data is if we show it always relative to the wild type channel. So for example, for changes in voltage in current density, as change of 0 means that it's equal to the wild type. There's no change in current density. When we look at the voltage dependence of activation changes, if it's 0, again, there's no difference to the wild type channel. So the data that I will be, wrong direction. So when the data that will be shown on the left will suggest loss of function while data results shown to the right of the line will indicate gain of function. Now, when I'm talking about loss and gain of function, I'm talking about channel function. We're not saying anything about neuronal function. Just want to make sure that that's clear. So these are the pulse currents recorded from the adult and neonatal NAV 1.6, where we're looking at wild-type channel in black, G1475 in red, and in 1768 in blue. We're looking at the peak current, the rate of inactivation, and the amount of persistent current that left at the end of this pulse. When we look at the properties, peak current, inactivation rate, and persistent current, you can see that for some of these properties, there's really not a major differences when you look at the adult isoform, which is open symbols, and the neonatal isoform, which is the blue symbols. But when you look at other properties, for example, peak current, depending on which isoform you're using, adult versus neonatal, now the variants are behaving differently. Similarly, when we look at voltage dependence of activation and voltage dependency of inactivation, when we look at the neonatal and compared to the adult, neonatal versus adult, for example, N1768, the behavior is very similar. There's no difference on which isoform you're looking at, being at wild type, the adult or neonatal. But when now, for example, for this other mutation, 1475R, now there's a difference in the behavior of this mutant, and it's dependent on which isoform is used. We also look at the frequency-dependent current decay. That is, when we are stimulating the cell multiple times, we're doing it at 20 hertz frequency here. We can see that in the adult form, the wild type and the 1475R mutant have a current decay very similar, while the 1768 variant has a much larger decay. In contrast, when we look at the neonatal isoform, both variants show a statistically significant larger decay than the wild-type channel. Also, when we look at the recovery through fast inactivation, that is, when the cell action potential go back to negative potential and the channel can go from the inactivated state to the closed state so it can open again, the wild-type and G1475R mutant for both adult and neonatal recovers at equal rate, while the N1768 mutant is much slower in its recovery process. In summary, wild-type and variant NAV 1.6 channels can be successfully analyzed by automated patch clamp. Adult and neonatal NAV 1.6 isoforms generate channels with distinct functional properties. The expression of epilepsy-associated variants in both adult and neonatal channels reveal that there are some changes that are specific to either adult or neonatal isoform that is used for the functional analysis or studies. And in conclusion, our results indicate that alternative splicing of NAV 1.6 is an important factor when assessing variant function, and that also automated patch clamp is a very successful high-throughput approach for determining variant pathogenicity. Thank you.
That was great, Carlos. Very, very interesting report. I had a question, and actually there's some feedback. I'm not sure if, can everybody hear me okay? Yeah. I had a quick question, and we'll look down the list in a minute as questions start coming in, but the time for the neonatal and the adult expression switch, you may have gone through that and I may have missed it. When is that in humans, do you know?
I'm sorry, so would you repeat the question, please?
Yeah, so the expression of the neonatal, when does that, compared to the adult isoform, when does that switch occur? Or is it?
Yeah, looking at the data, this recent paper, the switch occurs, let me see, I'm trying to, it's around, like maybe 2 months after birth. Okay, let me show the slide again, that way you can see that. If I'm allowed to do that, let me share the screen. So here you have, this is the adult and neonatal. And here, this is birth, half 1 year, 1 year after birth. And here, both of them are at equal level. But then the adult starts around 1 year, it starts being more than higher levels than the neonatal.
Great, thank you. Sorry, I missed that.
I have a question. Does this in any way help to explain why the people with epilepsy and loss of function tend to present with seizures later on?
If we we have some variants that are behaving differently between when we're looking at the neonatal isoform versus the adult isoform. So, I mean, if we want to take it to that level, yes, it will explain why the channel behaves. The same variant will give you one function early in development, but a very different phenotype later in development. That could be explained by this shift in the neonatal versus adult isoform and the distinct properties of the variants, depending on which isoform you're using as the backbone for the functional analysis.
So we have a great question from Andrew. Is it known whether different cell types have different relative expression of the two isoforms?
I'm sorry, what? My sound is really...
So, is it known if different cell types express relative different expression levels of the two isoforms? So, I mean, do certain neurons have more of the neonatal than the adult?
Yeah, I mean, for other sodium channels, it is known that that's the case. I'm not, unfortunately, I cannot give you a straight answer for 8A, but for other, like 1.2, it's known. For other, the voltage-gated sodium channels, that is the case, but I do not, I don't have an answer for 8A right now.
I think any more questions, John? Do you see anything? There's some coming up, but maybe in the interest of time, again, we may move on. But I had one last question I want to ask you that I think is really impressive, that your mutations, because that's something that's missing, right? Is is the activity of these SCN8A. So it looked like you were using your high throughput screen techniques. You're actually screening a lot of the mutations. Is that true?
Yeah, it is. I mean, it took a little while to get a channel, the DNA that is in a stabilized form where we can do the type of expression levels. But this year has been a little difficult year for everybody, so things were a little bit slow. But finally, we're ramping up, so we should be able to start pumping through more variants.
That's great. I think that information is needed. Thank you, Carlos.
Thank you. Thank you very much.
So now, as we move on, we're now going to have a talk by Ian Wenker, who is a postdoc in my lab. So I'll let Ian take it away. We can't hear you, Ian. I think we have a problem with the sound, Ian. Sorry. And you're definitely not muted because you don't have the mute button on. Any technical advice here? If while Ian figures out the sound, it was working. Can you hear me, Ian? Okay.
Could you first try just refreshing and coming back into it, see if that brings it back?
And if we continue having problems, what we'll do is we'll get Jeremy going while Ian figures out the sound. But we'll give him a minute just to see if we can get him back. I know it worked during the trial runs earlier, so.
Okay, can we hear me now?
Yep, you're good.
Yeah,
great. Thanks.
All right, now that you can hear me, yeah, my name is Ian Wenker. I'm a postdoc in Dr. Patel's lab. I want to say that I'm very grateful to be here and present this, as I think has been mentioned a couple times. We learn so much from the families and from the clinicians too. Also, I find it very inspiring. I've been in research for a little while now, and I've never really had such a strong connection to, the application of what we're trying to do, and it makes it feel really good. I also have to say that I've been very impressed in my short time doing this research and how well these mouse models, from what I've been doing, replicate what I hear coming from the family. So I think that's amazing. And today I'm going to talk about, on kind of the opposite end of the genetics from what Miriam was talking about, seizure behaviors that we witness, in particular behaviors relating to breathing and the cardiovascular system, mostly today about breathing. So, and what I'm calling, I have in quotations here in my title, tonic phase apnea, because it's not a term you'll find if you look it up. I'm kind of creating it, and hopefully I'll make some believers by the end. So briefly, background, the clear significance of this work is SUDEP, so sudden unexpected death in epilepsy. This is, a very difficult topic, but a very important topic. It's been a focus area at the NIH for a little while now, and over the past decade or so, there's been more and more research done on the topic, but we still don't really understand the mechanisms of SUDEP. The U stands for unexpected, but it might as well stand for unexplained. We think in the field.
Can't see your slides. Are you sharing your slides?
Yeah, I'm sharing. You can't see them?
No. Can anybody else see them?
Can you see them?
No.
If I go to the out of presentation mode. Oh, I'm sorry. I want to do entire screen. I want to share audio. Oh, I see.
I've just got the images of it.
I think, yeah, I forgot to click.
There you go. It's working now. Great.
Sorry about that.
That's okay.
It's good timing. It's just the title slide that you missed. So there's the title slide. So some background. The significance is sudden unexpected death in epilepsy, or SUDEP. And so it's unexplained, and we're trying to understand more about it. What we do know, or what we believe, is that it's due to a failure of either breathing or cardiac activity. This is because of the sudden aspect of it, and these are the two systems that promote life in the short term. Now, making recordings of these systems in patients is extremely impossible, and SUDEP is uncommon enough that would be very difficult to do. And that's where these wonderful mouse models come in. As Manoj mentioned earlier, these mice, we're not treating these mice. In fact, they have to undergo some invasive surgeries by me in order to do these recordings. And in addition to that, we can just breed many mice and record even a rare event we can observe by just doing many recordings at the same time. I'm going to present data in a short time from 3 mouse models. I'm not going to get into the genetics, but just to say that we have a model where we can look at spontaneous seizures that are of different kinds, and only one kind we find actually produces SUDEP, so we can learn from that. And then 2 other mouse models where we can induce seizures with sound, so audiogenic seizures. And this allows us to then intervene and try to understand what's going on in a more causal way. It's a short talk, but I like outlines, so briefly, I'm going to show you a little bit about the methods that I use. I'm going to try to convince you that breathing, or lack thereof, is really what produces SUDEP in these mice. Then I'm going to talk to you about the tonic phase and how that relates to SUDEP, and how the tonic phase relates to apnea, hence my tonic phase apnea. And finally, show you data on what we think is the potential mechanism that produces this apnea, and some future studies to try to understand that better. This is a cartoon schematic that I use to demonstrate the kind of recordings that we're doing. We can record EEG, ECG, and breathing chronically in these mice. EEG and ECG are through pretty standard methods with a headset on the mouse that's connected to a cable through a swivel, so the mouse can live in its home cage. We connect a pressure transducer to the chamber to sense very small changes in pressure that tell us what's going on with breathing. If we expand out this data and look at it, we can see in the ECG signal, QRS complexes. This indicates heartbeats, and so we can get cardiac frequency and see if there's a bradycardias or arrhythmias. There's also some muscle artifact in these signals, which at first was, is disappointing, but actually it gives us, as you'll see in some of my data, a very clear depiction of when the tonic phase is happening, which I think is very important. The blue traces are breathing, so these small changes in pressure are individual breaths the mouse is taking. So downward is breathing in, and upward is breathing out. So we can get breathing frequency, breathing amplitude, and when there's a flat line, it means that there's no breathing and we're seeing an apnea. So now I'm going to show you a fatal seizure in one of these mice. This is all kind of cropped down to make this video. So we're actually recording from a number of mice at the same time in order to capture this event. And so again, in blue, we have breathing. In red, we have ECG. And then in black, we have EEG. I'm going to push play. And we're looking at the mouse in the red box over here. The seizure starts pretty much immediately. The mouse is going to have some convulsions during the first half of the seizure. Breathing is still occurring. It's actually increased, as well as heart rate. The mouse will go through a wild running phase that ends in a tonic phase. And so you can tell it's a tonic phase because of their posture and because of this large amount of muscle activity in the signal, in the ECG signal. And at the same time, we see a flat line in this breathing signal. So there's no breathing going on here. If I connected this pressure transducer to an empty chamber with no mouse, it would look like this. At the end of the tonic phase, we start to see that there still are heartbeats. It's slower, but it is occurring. But there's no breathing. And if we draw this out 15 or 20 minutes, we'll see the heart will eventually stop, likely due to lack of oxygen, so the cells can't function well anymore. And we've seen this in several mice now, and what we believe this means is that apnea happens first, and it is the causal event that's producing this SUDEP. If this is true, then if we were able to reinstigate breathing through a ventilation, mechanical ventilation, we should be able to rescue this phenotype. So this is a different mouse model where we found that we can induce seizures with this device that makes a kind of high-pitched noise. And that at this developmental time point, these mice, they do not survive these seizures. So most of these seizures are fatal. And so I'm going to have one mouse where that happens and one mouse where I show you when we ventilate them. So we turn the sound on. The mice have a seizure pretty much immediately. They go into this tonic phase. You can see their legs are extended. And this mouse on the right, I ventilate them with my fancy ventilator. And then on the left, I handle the mice and do a sham ventilation where I don't attempt to ventilate them. They're in the tonic phase. Eventually, the muscles will relax in this mouse on the left. You can see the ear go up, but they won't start breathing again. On the right, I keep ventilating them until I see that they're breathing on their own, and then I stop. And this mouse will be postictal for a little while, but they will recover. A few hours later, if you see them with their brothers and sisters, you wouldn't be able to tell which one had the seizure. So they recover well. So giving them artificial ventilation during this narrow window of time is enough to rescue the SUDEP event in this mouse. So now I'm going to switch over and talk a little bit about the tonic phase briefly. The tonic phase is important for SUDEP in this mouse model we find. Of all the spontaneous seizures were recorded, only tonic seizures are fatal. The clonic seizures that lack a tonic phase are not fatal. Not all tonic seizures are fatal, as you can see from the plot, but some are. And I just want to show from a paper this year, looking at 4 other mouse models, non-SCN8A, that this also seems to be true in some other models. So this might be a little bit generalized to a few epilepsies. I think the tonic phase is essential for apnea, and what kind of led me to this conclusion is a few things. First off, the apnea and the tonic phase happen at the same time. This is examples of a fatal and a non-fatal seizure. We can see this tonic phase here and here, and we can see the flat line in the breathing in both cases. Even in a non-fatal seizure, they have a prolonged apnea during this time that just eventually recovers. So that's 1. 2, in clonic seizures, there's no apnea. In fact, their breathing frequency increases. And so we see this very clear grouping of tonic seizures and clonic seizures when it comes to breathing during a seizure. And also within the tonic seizures, the duration of the tonic phase actually correlates to the duration of the apnea. So a longer tonic phase produces a longer apnea. And so they're related in that way too. So I think there's some mechanistic connection between these two events. So lastly, I want to describe a potential mechanism. And to do that, I want to describe just some very quick and basic breathing mechanics. So in humans, at a minimum, to breathe, you need inspiratory oscillation. And so what that means is if you recorded from the diaphragm, we would see, that's not playing, that when the diaphragm contracts, we breathe in, and when it relaxes, we breathe out. And so you get this oscillatory motor activity. You can produce apnea in two ways. You could have no motor activity in the diaphragm, which you'd have a flat line, or you could have a tonic contraction of the muscle. This would be something like a inspiratory breath hold you do when you jump into a pool or into water. Since the tonic phase is noted for having a lot of muscle activity, I hypothesized that it was due to tonic contraction. And I moved forward to do recordings from D+ mice, so different mutant mice, because they have seizures when they're older, so it's a little easier to do these experiments. I'm also able to induce audiogenic seizures in these mice. Recording from the diaphragm looks like this. We see bursts of electrical activity in the diaphragm that correspond to inspiratory breaths. You can see them here, and when we have a larger breath, we have a larger amount of diaphragm activity, as one would expect. In this mouse, we can now induce an audiogenic seizure. The first half of the seizure, again, is noted by a high frequency breathing, which we can still see this oscillation in the diaphragm. It's just faster. Then during the apnea, we see a tonic contraction of the diaphragm. In fact, a massive amount of contraction here, and we lose this rhythmic oscillation that we usually see. And so I would say that this contraction of the diaphragm is sufficient to produce an apnea. I can't say that there aren't other things going on at the same time, but this is certainly not good for trying to get normal breathing going. And so right now we're gearing up for a couple sets of experiments to try to understand this better. So sort of the way the research has worked so far is we started at the end, what happens in these events, and then we're starting to work our way back. We looked at muscle, and now I'm going to go back to the brain. And I want to record from what are called upper motor neurons. These are neurons that project to the spinal cord to activate muscle activity. This picture just shows one, the primary motor cortex, but there are several others. If we could figure out which ones are activated and causing this diaphragm contraction, we may be able to intervene and change how that happens so that we don't have apnea. Another set of experiments that we're really excited about in the lab are using some known circuitry in the brainstem to try to intervene. When you breathe, the rhythm that causes you to breathe is generated in your brainstem. There's an inspiratory oscillator in the ventral brainstem, and it's regulated by several neuronal groups that I've worked with in the past. And so there are some that can inhibit the inspiratory oscillator and some that can activate it. If it's tonically active, what we want to do is inhibit it. And so we're going to use what are called optogenetics to stimulate this region called the Botzinger complex and inhibit that to try to recover breathing. Similarly, we could try to stimulate breathing and see if that can recover breathing after the tonic phase, when we expect that the inspiratory oscillator may be not functioning. It might not have enough input to function properly. So we're very excited for these future directions. Hopefully next year I'll have more to talk about on these. And so I want to thank all of you for paying attention, and I take any questions if I haven't gone too long.
We are running quickly out of time, but there is one here from Alex. Ian, where is the origin of C3, C4, C5 that keeps the diaphragm alive?
The origin of, so the phrenic nerve that innervates the diaphragm comes from those segments of the spinal cord, C3 through C5.
Yeah.
Both in humans and in mice, I mean.
Yeah.
So those are your cervical spinal cord sections, and I'd probably get the actual numbers wrong, but they're a little below your neck. I think they're fairly they're up higher in that region.
Okay, great. They're on the screen, but then we should move on to Jeremy now. So Jeremy, you're on.
Okay. Can everyone hear me?
Ian. Go ahead, Jeremy.
All right. I'm trying to share the screen. See if that'll work. Okay, can everyone hear me?
Yep, we can.
All right, awesome. Cool. All right, so thank you everyone for the opportunity to speak here. As Manoj said, I'm Jeremy Thompson. I work in his lab. I'm a PhD student. And I'm going to talk to you guys about the role of a cell type that we don't really talk about too much in these meetings, and that's astrocytes and how they play a role in SCN8A epileptic encephalopathy. And hopefully by the end of this presentation, I can convince everyone as to why we should care about astrocytes in seizures and epilepsy, a disease that I guess we can argue is mostly about neurons. So to frame why these cells are important, I want to give a little background about these cells before I go into my data. So astrocytes are a part of a group of cells called glial cells, and they have a whole host of very diverse functions in the central nervous system. One of their functions is barrier formation. So we're all familiar with the blood-brain barrier, and these are one of the cell types that contributes to that. Here's a cross-section of a blood vessel in the brain, and astrocytes actually send their little processes, we call them in feet, to wrap these blood vessels. And they also form the inner lining of the brain. You can kind of see that highlighted in green up here, and this is a mouse that expresses a green protein in its astrocytes. And so this sort of has a protective function for the brain overall. They are also involved in neural communication. So we throw out the word tripartite synapse. So a synapse is where the two neurons interact, but the third element of that is the astrocytes, and they are involved in transmitter recycling. So each time a neuron communicates to another, it releases neurotransmitter, but in order to attenuate that signal and allow good fidelity of neural communication, this neurotransmitter has to be removed, and it's the job of astrocytes to do that. And importantly, for this talk, is that astrocytes also contribute to ion homeostasis. And I'm going to show some data that I have on astrocytes and how they regulate potassium in SCN8A. And each time a neuron fires, it releases potassium. And astrocytes need to take this potassium up, and they do so through these potassium channels. So in various cases of insults to the brain, astrocytes take on this different shape and characteristic, and we call them reactive or gliotic. And these reactive or gliotic astrocytes, astrogliosis, actually interferes with their normal functions. So typically we have a resting astrocyte in a healthy brain, and it has a certain shape. It's got a small little cell body and these skinny little arms. But following an insult to the brain, my animations are not, okay. Following an insult to the brain, and that insult can come in many different flavors. It can be an infection, it can be a traumatic brain injury, or it could be a genetic insult that leads to seizures and epilepsy, as in this case. The astrocytes will become reactive, and it's defined by a shape change. So their cell body will become bigger, their processes will be thicker and stubbier, and this is really important because it gives us an indication that the normal functions of the astrocytes are performing are impaired. And this is because of a downregulation in some of these proteins. So for example, the potassium channel I'm going to talk about, as well as neurotransmitter transporters that are essential for them to carry out their functions. There is also an increase in their expression of this cytoskeletal protein called GFAP. And I'm going to be using that in my data just as a marker of astrocyte reactivity. And when I looked at the brains of our SCN8A model mouse, we're using the N1768D model that Miriam Meisser's group developed. This is what I see. So in green is the GFAP, that marker of reactivity, and these are spontaneously seizing mice. This is in the cortex. On the left, I have a wild-type healthy mouse, and then a mouse heterozygote for the mutation. There's an obvious increase in GFAP, indicating that these astrocytes are reactive. We can zoom in on these guys to appreciate their change in morphology. We have these nice little thin, delicate-looking processes in the wild type, which become thicker, more stubbier processes in the seizing mutant. And we can quantify this percent area increase, and there is a significant upregulation of GFAP. Now, when I show this data, I always get the question, well, are there more astrocytes in the D+ mouse? Because that's what this looks like, but that's actually not the case. These individual astrocytes are increasing their expression of GFAP. And I can tell this because I have another way of looking at these astrocytes in tissues. So I have this, we call it a reporter mouse. So this ALDH101 is a gene that astrocytes express. And we can force cells that express this gene to express a green fluorescent protein. And so here I'm visualizing all astrocytes in the cortex, not just those that are reactive. And when I count these, there is no increase between the two groups. And so these mice have this mutation from the moment they're conceived through adulthood. And one of the questions that I had was, are astrocytes dysfunctional earlier, before the seizures arise, or do astrocytes become reactive in consequent as a result of these seizures? So I can look at mice that are pre-seizing. So these are younger mice; they're only 3 weeks old, and I can look in their cortex and stain for my astrocyte reactivity marker, and indeed see that there's no increase in the percent area covered by GFAP, indicating at a time when these mice are not seizing, the astrocytes are not reactive, suggesting that this reactivity is potentially caused by the seizures. Although I do have some future directions to get at this in a more granular way, because right now we have just snapshots at these different time points. So as I've stated, astrocyte reactivity is often, not always, but is often associated with deficits in their normal functioning. And the one that I've chosen to look at is the potassium regulation through the potassium channel KIR 4.1. And so in astrocytes, they are the main expressors of KCNJ10, which is the gene that encodes the protein KIR 4.1. This is looking at the expression of this gene in astrocytes compared to the whole tissue from data from Bob Cox's lab. And this is important because, as I mentioned, each time a neuron fires an action potential, it releases potassium into the extracellular space. And if there's too much extracellular potassium, this will cause neurons to become depolarized. And a depolarized neuron, it's more easily stimulated to fire an action potential. And so we can consider them hyperexcitable, which, along with hypersynchrony, are two of the hallmarks of seizures and epilepsy. So by astrocytes taking up this potassium, they are actually acting to dampen neuronal activity and be sort of anti-epileptic. So it's already been demonstrated that global loss of KIR 4.1 results in seizures and early mortality in mice, giving us a good impetus to look at this channel in our mice. So I've done patch clamp electrophysiology on astrocytes in brain slices from the wild type and confirmed seizing D+ mice. So just like for the immunohistochemistry data I showed you, all these mice were video monitored and had surface EEG, so we know their seizure history. We can measure the total current across the astrocyte membrane, and it's in fact decreased in our seizing mice, and it's demonstrated here by a current and voltage plot showing reduced current overall. But we have a small molecule, barium chloride, that inhibits the KIR 4.1 channel. And when I apply that blocker, measure the current, I can subtract out my KIR 4.1 sensitive current and see again that the D+ mice do have a reduction in KIR 4.1 mediated current, shown here in a current voltage plot, where the orange line is below the blue, so less current is being shunted through this channel. So this indicates that astrocytes, through their inadequate clearance of potassium, may contribute to the seizure phenotype that we're seeing in the N1768D mice. And it's just summarized here, where overall astrocytes become reactive in seizing D+ mice. And in these mice, they have reduced potassium currents. But we really want to get more granular and figure out when exactly do these astrocytes become reactive? Is it that astrocytes become reactive before seizure onset and contribute to the development of these seizures, or are they simply responding to the aberrant neuronal activity? And this is a ongoing / future experiment. And the way it works is we're able to visualize the astrocytes in the living mouse tissue. So we're doing chronic imaging of astrocytes via cranial window implantation. So it's a surgery where we actually remove a piece of the skull, put a glass cover slip on, some dental cement, and then let the mouse recover. And then using that astrocyte reporter mouse that expresses GFP, I'm able to take the same mouse, video monitor it 24/7 so I know when it seizes, but I can go back and look at the same astrocyte in that living mouse week after week, and I can correlate the changes I see in astrocyte shape with the seizure history. So really excited about that. And so hopefully I stayed under my 10 minutes, but I would just like to acknowledge the Battelle Lab and everyone who has helped me, and as well as some collaborating labs. And with that, I will take some questions.
Jeremy, that was a great talk. I think I've got some replay. And Andrew, I don't know how much time we have, but maybe just for 1 or 2 questions. Andrew had a question. His question is the astrocyte change specific to SCN8A dysfunction or a general consequence of seizure activity?
So there are other models. So in the canic acid model, astrocytes become reactive. In the pilocarpy model, astrocytes become reactive. So it's not necessarily specific to SCN8A.
Okay. And JC, or no increase in GFAP mRNA level prior to seizure. GFAP mRNA levels only increase after seizure. So that's kind of in agreement with what you're showing. Which is
yes.
nice.
Yeah. So during the presentation, I did emphasize seizing D+, and I do have data showing that in some of the mice that are old enough to have seizures but haven't yet, I don't see the increase in GFAP expression.
Okay, and GFAP went up within 24 hours of seizure onset in D+ mice. That's JC.
Okay.
Maybe one other question. We have transcriptome data from mice at 56 days pre-seizure and after 5 seizures and 20 seizures. This is from Michael Hammer, so that will be interesting to see if he sees a difference in the astrocyte markers. Okay, so I think that's all on there for now. So I think this is the time when we open it up for more discussion, Casey and Hillary, or have we run out of time?
I think we may have run out of time, but just an FYI, you guys can keep with the chat, because the rest of the evening will be on this session. You guys can absolutely keep going with the chat discussions.
Okay.
Thank you.
Okay. So with that, thank you to all. Sorry, I was just going to say with that, I'd like to say thank you to all the speakers. That was a lovely session. I really enjoyed it. Thank you.
Absolutely, 100% agree. Clinicians or researchers, thank you for not only participating in our roundtable today, but really valued our children. As parents during these discussions, so our hearts hope and on this fight against SCN8A. So thank you.